want to thank the organizers for giving me the opportunity to present our work. So today I'm going to present our project on studying how the influenza virus initiate innate immune responses in human volunteers, human vaccinees. As we know that influenza vaccines, particularly the trivalent inactivated influenza vaccines, is among the most successful vaccines. And it can elicit effective protective immune responses in most of the vaccinees. So, so the major protective arm of immunity is the antibody responses, of course. But it has been shown that the TIV can also induce very good helper T cell responses. And even TIV is an inactivated virus, it can still initiate quite potent cytotoxic T cell responses. And the CD8 T cell responses actually are responsible for protections against influenza infections in certain contexts, such as in elderly people or during the 2009 pandemic, influenza pandemics. So, so we want to use TIV as examples to learn how the effective immunity can be induced by a successful vaccine such as TIVs. So in order to induce neutralizing antibodies that can block viral infections or induce CD8 T cell responses that can kill virally infected cells, the adaptive immune responses has to be regulated or even dictated by the innate immune responses. And among innate immune responses, the dendritic cells, which is play the central roles in regulating not only the innate immune responses, but also the adaptive immunity. So it kind of, so dendritic cells serve as the bridge that link the innate immunity and adaptive immunity. So we want to know what TIV has done to the innate immune responses that can subsequently initiate very effective adaptive immune responses by the TIV. So dendritic cells are the central regulators of immunity that bridge innate and adaptive immune responses. And it is the principal professional antigen-presenting cells in the immune system. It can capture, process, and present antigens to, and in order to trigger an activation and the differentiation of antigen-specific CD8 and CD4 T cells. And dendritic cells are unique because they are the only antigen-presenting cells capable of inducing primary responses of naive CD4 and CD8 T cells for activation, proliferation, and differentiation into effector and memory T cells. But more importantly, dendritic cells are the only type of cells that can undergo cross-priming of naive CD8 T cells through a unique cross-presentation of exogenous antigens. So we want to know whether or not TIV can actually generate in vivo that of the cross-presenting dendritic cells in order to initiate CD8 T cell responses. And so the blood monocytes are the precursors of dendritic cells. So under different conditions, monocyte can be driven to differentiate into different types of dendritic cells, either under steady-state conditions or under various inflammatory conditions. So the specific aim of this study is very simple. We want to investigate the effect of TIV on the phenotypes and functions of blood monocytes at early time points post-TIV immunization. So the study protocol is also very simple. So we recruited 20 healthy volunteers. They are going to receive seasonal influenza vaccines shot, TIV injection. And then right before TIV injections or at different days, including 1, 2, 3, or 5 days post-TIV shot, we collect the blood and isolate the monocyte and then study their phenotypes and functions. So the monocyte isolated either before or after the TIV shot, we analyze first the surface phenotype of these cells. And surprisingly, we found that immediately after the flu shot, we can see the significant downregulations of many macrophage-associated markers, such as the scavenger receptors CD163 and CD36. And also, they are both mature macrophage markers. So we see a very rapid downregulation of these markers. And also, we did intracellular staining for different LAMP markers. So CD68 is the macrophage-associated LAMP, which is also the expression of CD68 is also downregulated very significantly upon TIV injection. But however, the other LAMP, CD208, which is dendritic cell-associated markers, is not affected by TIV immunization. And also, strikingly, some markers that never been seen on the surface of monocytes or even cultured monocyte-derived dendritic cells will start to be expressed on the monocyte of TIV after TIV injections. So this includes the CD1B, which is a non-classical MHC molecules. So you can see after 1 day of TIV injection, many of the almost all the vaccinees, their monocytes start to express CD1B, and also this chemokine receptor CCR6, which is also a unique marker for dendritic cells. And also another co-stimulatory molecules, 4-1BB ligand, is also triggered, the expression is also triggered by the TIV injection. And also, we also found the upregulation of CD40, CD86, and CD80s on the monocyte induced by TIV injection in the volunteers. So another hallmark of dendritic cell is that they are able to produce potent inflammatory cytokines. So we performed intracellular stainings directly on the freshly isolated monocytes to see whether there are cytokines already produced inside those cells. And strikingly, we found that shortly after the TIV injections, so the monocyte isolate before the immunizations, there is no TNF-alpha production under the steady state. But however, shortly after TIV injections, we start to see there are abundant amount of TNF-alpha present in the monocyte that is freshly isolated from the volunteers. And we can see the production of TNF-alpha in the monocyte in 5 of the 7 volunteers. So this gives us some hint that TIV injection already triggered the monocyte to express the phenotype of dendritic cells and also production of pro-inflammatory cytokines. So another hallmark of dendritic cell is the is to stimulate the primary proliferative responses of allogenic naive CD4 and CD8 T cells. So we performed a mixed lymphocyte reactions by co-culture the purified monocyte, isolate at different days before or after influenza immunizations, also with the naive allogenic naive purified CD4 positive or CD8 positive T cells. And we found that the monocyte isolated before the influenza immunization, they have no stimulatory activity. But however, the monocyte isolated shortly after the influenza immunizations, they start to show the ability to stimulate the proliferation of allogenic naive CD4 and CD8 T cells. And their capacity is pretty much similar to the cultured dendritic cells. So that means shortly after influenza immunizations, the monocytes start to acquire the ability to stimulate allogenic T cell responses with, to the extent similar to the cultured dendritic cells. So we studied those CD4 or CD8 T cells that have been co-cultured with the TIV-induced monocyte to see whether they have changed their phenotypes. And it is indeed the case. So we found that those CD4 or CD8 T cells co-cultured with TIV-induced monocyte, they start to downregulate CD45RA and upregulate CD45RO, and also downregulation of CD62L and or CCR7 and upregulation of Fas ligand. So this shows that those T cells co-cultured with the TIV-induced monocytes start to express the phenotype of memory T cells. And the ability is quite similar. The TIV-induced monocyte is quite similar to the culture disease in triggering memory T cell development. And then we study whether those differentiated CD8 T cells has the ability to conduct certain effector functions. So we found that the CD8 T cells generated in the co-culture with monocyte derived before the flu shot, there is no killing by those CD8 T cells. However, if the CD8 T cells, if they were co-cultured with the monocyte derived after the TIV injections, we found that those CD8 T cells were able to kill target cells in a killing assay. So that means the influenza vaccines induced monocyte are able to induce T cell with proliferation, differentiation into memory cells, and also effector T cells. And then we analyze whether those T cell, TIV-induced monocyte can efficiently capture antigens, and it is indeed the case. So the monocyte isolated after the TIV shot, they have enhanced ability to uptake dextran. But unlike cultured dendritic cells, they capture the antigens through a mannose receptor or DC sign independent uptake, which is also trypsin resistant, but PI3K dependent. So that means those TIV-induced monocyte has a unique ability to uptake antigens through macropinocytosis. So we want to know if they can capture the exogenous antigen, we want to know whether they can cross-present those exogenous antigens to stimulate CD8 T cell responses. So we fed those TIV-induced monocyte with different antigens, including tetanus toxoid, flu antigens, or PPDs or BCGs. And we found that not the steady-state monocyte, but the monocyte isolated after the flu shot, they were able to present those exogenous antigens and stimulate the proliferation of autologous naive CD8 T cells. And this cross-presentation can even be enhanced by the presence of some co-stimulation such as TLR ligand, cytokine cocktails, or interferon alpha. So in conclusion, the TIV-induced monocyte isolated 1 day after or 2 days after TIV shot can induce efficient cross-presentation of exogenous antigens to prime naive CD8 T cells in a T helper cell independent manner. So that may explain why the influenza, although it is an inactivated vaccine, but it can still stimulate the CD8 T cell responses in many of the vaccinees. And then we studied there are two different subsets of blood monocyte. One is CD16 negative, and the other is CD16 positive monocytes. So CD16 negative monocytes are the classical monocyte, whereas CD16 positive monocytes are inflammatory monocytes. So we want to know whether these two subsets of monocytes are differentially modulated by the TIV vaccine. So we separate different subsets of monocyte when we isolate the monocyte from the vaccinees. And surprisingly, although the total monocyte do not have the stimulatory ability on the steady state. But however, the separated CD16 positive monocyte has had already the stimulatory activity even without TIV vaccinations. So this surprising finding suggests that the CD14 positive, CD16 negative monocyte may have a suppressive effect on the CD16 positive monocyte in terms of stimulating T cell responses. However, after TIV immunizations, you can see that both of the subset of monocyte acquire the ability to stimulate T cell responses. So that means TIV may have lifted the suppressive effect of CD16 negative monocyte, and in turn, they can, so the monocytes start to acquire the ability, such as the cross-presenting dendritic cells, to stimulate T cell responses. And also these two type of monocytes, CD16 negative or CD16 positive monocyte, they induce the differentiation of memory T cells in different ways. So in brief, we found that CD16 negative monocyte, which is the suppressive regulatory monocyte in terms of influenza immunizations, they actually induce CD45RA intermediate, CD45RO low, IL-7 receptor alpha positive, CCR positive effector memory T cells. Whereas the CD16 positive monocyte, after TIV immunizations, they can induce the CD4 T cells and CD8 T cells to differentiate into central memory T cells. So that means influenza, the TIV vaccines induce CD16 positive monocyte and negative monocyte in different manners. So we want to know whether this vaccine-induced monocyte differentiation into dendritic cells is the direct effect of the vaccine, or is because the vaccine induced something else that can in turn affect the differentiation of monocytes. So we isolate monocyte in different days, and then we culture the monocyte with the sera isolated on the same day, overnight. And we found that if the monocyte isolated after 1 day of TIVs and then cultured in the presence of the sera obtained from the same days, those sera can actually further promote the DC differentiation after overnight cultures. So that means the induction of monocyte to DC differentiation by TIV is actually mediated by the vaccine-induced some serum factors. So we guess what type of serum factors may be responsible for TIV-induced monocyte to dendritic cell differentiation. So the first guess would be the type 1 interferon, because influenza virus and particularly TIV induce potent interferon production in host cells. So to this end, so we recruit 6 hepatitis C virus infected patients who are treatment naive, and they are to be injected by interferon alpha. So we collect their blood monocyte right before interferon alpha injections, or at 1 day after interferon injections. And we found similar patterns of monocyte changes. That means after interferon alpha injections in those patients, we found that downregulation of macrophage markers on the monocyte, but they start to express certain unique dendritic cell markers on the interferon alpha-induced monocytes. So in conclusions, we found that TIV injections can rapidly induce the differentiation of monocyte into cross-presenting dendritic cells, which can stimulate the proliferation, differentiation, and effector function development in naive CD4 and CD8 T cells, either in allogenic reactions or in autologous antigen-presenting assays. And they produce cytokines, and they can capture antigens through a very unique mucopinocytosis pathway. And this differentiation triggered by TIVs appears to be mediated by soluble serum factors elicited by the vaccines. And also, interferon alpha administration in patients with HCV infection induced a similar programs of monocyte to DC differentiation as seen in influenza vaccination. So I thank my group members, Kai-Feng and Hui-Ru, for their hard work. So thank you for your attention.
Any questions for Dr.
So you mentioned the PI3 kinase dependency of the reactions, and how that could you explain a little more about the PI3?
Okay, so dendritic cell is known for, they are very efficient in capture antigens for antigen processing and presentation. So the data from the, most of the time we study dendritic cells by culturing the monocyte in the presence of IL-4 and GM-CSF, which is, I think, is very artificial. So for those cells, they capture antigens mainly through like mannose receptors or DC sign-mediated internal endocytosis. But however, we fed those cells with the same antigens, dextrin, which is a carbohydrate antigens. But we found that we cannot block the endocytosis by using the blocking antibodies to DC sign or mannose receptors or mannans. And also, if we treat the cells with trypsins, they can still uptake those antigens. But however, the antigen uptake can be inhibited by the PI3K, and that is a typical sign of mucopyanocytosis. Means they just use the fluid phase uptake of those small antigens. So it is receptor independent.
So, any question? Yeah, go ahead.
I didn't listen clearly enough. Were these PDCs or MDCs? Which subtype or subclass of DC cell are you referring to? CD11C positive or not?
Okay, so they are, CD11C are expressed in humans. So in human cells, even monocytes express CD11C. So it's difficult to use CD11C as the marker for dendritic cells. But however, we do find some markers such as so CD1B and CCR6 are the unique dendritic cell markers in vivo. However, when people use cultured monocyte-derived dendritic cells, they do not express these kind of markers. So this marker can only be detected in dendritic cells in vivo, in situ. So I think this type of TIV-induced monocyte is more like really bona fide dendritic cells than the cultured monocyte-derived dendritic cells.